Everyone. My name is Dalibell Ferreira, and I'm from the class of 2007. I'm enrolling in Harvard College in the fall. And my other group
I'm Sheryl Todman, and this is my first year at UCS, and I go to Young Women's Leadership School, also known as Nunny, Reyes, also 12. I'm Tashneen, and also 12. I'm from Marroquin, and yeah, I'm also 12. Jessica Sinchi, I'm also 12.
I'm St.Ange. This is my second year. I go to Manhattan Hunter Science High School.
And our other group member, Sadat, he cannot make it today. Our project is called Biodiversity and Nutrient Cycling in Piermont Marsh. And our mentor is Dr. Bakht, and we studied at the Lamont Doherty Earth Observatory, and that was part of Kalib University. So our background on our research is that we studied in Piermont Marsh, which is located 20 kilometers north of New York City on the Hudson River. Over there, we observed habitats in the erosional depositional banks, the rivulets, and the pond, which is located inside the marsh itself. We also studied the Phragmites australis, which is an invasive species in the marsh, and we studied its effects on the hydrology in Piermont Marsh. We also observed the aquatic species of the Piermont Marsh. So, this is our study site right here. And the thing with Piermont Marsh is that it has a lot of creeks and channels, and it also has ponds within. So we studied in Squirrel Creek, Tidal Creek, and Concord Creek, as well as the Hudson River Channel, which is right here. And we also studied the interior pond, which is inside the marsh.
Our objective was broken down into two parts: biological census, what makes animals use the marsh when they do, how site location affects the number and species of the fish, and hydraulic sampling. We tested the phosphate, nitrate, and nitrite levels in marsh, and we also wanted to see how spring and neap tides affected the nutrient cycle. How does Phragmites australis alter the marsh functions? One way it alters the marsh function is by raising the substrate above the water level, which means the fish can't get to their nutrients. It also raises the elevation of the mud, so that controls how often it's flooded, like how many times a month, and how long. Although Phragmites australis is dominant in the marsh, there are several indigenous species to the marsh, such as Hibiscus, Petralis, Spartina patens, Spartina alterniflora, and Discus specifica.
Okay, so for the basic field materials in the marsh, we use 48G minnow traps, which are right here. We use them in sets of 3, and 48-meter cotton rope to hold them together. 16 1-meter lengths of 3/4 of an inch of PVC pipe, which is right here, and we use that to locate our sites. And we also use a line side, and it's basically an instrument around this size. And we use it to measure conductivity, salinity, temperature, and dissolved oxygen. We also have clipboards and data sheets to record our data, a GPS system, 6 plastic containers that we put our fish and invertebrates in, and a formalin solution that will preserve and kill the animals. And we have gloves, and we use tape to label our sites according to whether they're a rivulet, erosional bank, or depositional bank. And these are our lab materials. Gloves again, a microscope, isopropyl alcohol 50%, fish and invertebrate keys, 6-inch metric ruler, electronic balance, sampling syringes, filter holders, filters, water sample bottles, sampling tubes, a portable ice store that we use to preserve the samples, and a micropipette and beakers.
Okay, so to go more into what we do in the field, we go out with PVC pipes and mark the erosional, depositional, the rivulets, and the interior, and the channels and the ponds. So to go deeper into that, we attach 3 minnow traps, and that's one set, and we attach it to the PVC pipe, which is right here, and if you notice it's red and yellow. Red is for erosional and yellow is for rivulet. Then we measure the water length of the PVC pipe, which is marked in centimeters. When it's in really high tide, it can reach about 100 centimeters, or it can reach at 0. Then we use the YSI to measure temperature, salinity, and dissolved oxygen. And with the fish we capture, we put them in jars, and we put them in 10% formalin. And after 24 hours, we rinse the formalin, and we put them in 50% isopropyl alcohol. And what we do once they're in the alcohol, we take the fish, we have a ruler, and we start measuring them. We measure them in millimeters, so we can get fish as big as 35 millimeters or 90. And then we would have eels, and those have to go in a separate category, which would not be weighed, and the catch.
Can you hear me? All right. We also had a second part to our presentation that included the lab work. And in the lab, we had water samples from the marsh. And with those water samples, we would test for nitrate. And nitrate is very harmful to the fish. And the reason is because of eutrophication, which I will explain right now. It's a process when there's an overload of nitrate from the cities. And it's a really great fertilizer to the plants. And when they have a great bloom, they eventually die and rot. And these dead plants will consume a lot of the oxygen in the water. And this can literally suffocate the fish, which will greatly affect the hydrology of the marsh. So we have our samples, and we use a pipette to transfer 10 milliliters of it into a separate test tube. And to achieve reduction, we will use a cadmium bed column to achieve the reduction of nitrate into nitrite. We also tested for nitrite, and we used a pipette. We transferred 400 microliters of the sample into each well. We added 25.2 microliters of soap, phenylamide, and mixed it for 30 seconds. We had to wait for 30 seconds so that the two solutions will properly mix. We also added 25.2 microliters of dihydrochloride and mixed for 30 seconds. We waited so that they can also mix again. We recorded the data using a digital spectrophotometer and absorbance program. The way a spectrophotometer works is that the samples that we had were put into cuvettes, and these cuvettes were put into the spectrophotometer, and we would place the cuvettes with the shiny side. Some of them have a dull side. We place the shiny side. It's horizontal to the machine so that the light will pass through it. The more light that the cuvette absorbs, that means that there is more nitrate present in it. And nitrate is a nutrient, for those that don't know.